Hello and welcome. This is Dr. Teresa, and in this lecture, we're going to start into the nervous system. So we're going to talk through the anatomy and physiology and the divisions of the nervous system. We're going to describe the three functional classes of neurons. We'll describe the soma, the dendrite, the axon hillock, the synaptic bulb, the myelin sheath, and the nodes of Ranvier. We're going to compare and contrast neurons with the structure and function and location of neuroglia. So the nervous system, if you've not sat down and actually thought it through, really is a big like 911 dispatcher of the body. It receives information, it interprets and integrates the information, and then sends out a command or a direction for a part of the body. So it does this by way of coded messages. The coded messages can come via electrical charge or chemical packet. And when we're talking about this, we are talking about this at the cellular level. When we talk about the electrical charges and impulses that are the messages of the cells, this goes down into the action potential across the cell membranes that can be created. This goes into the polarity differences from the inside of the cell to the outside of the cell. When we talk about the chemical packages that are sent between the cells in the nervous system, we are primarily talking about neurotransmitters. The way that we break down the nervous system is into two primary divisions. We have the central nervous system and the peripheral nervous system. The central nervous system is comprised of the brain and the spine, the brain housed within the skull, the spine housed within the vertebral column. The peripheral nervous system then is anything outside of those two, outside of the brain and spine. Now we have some structures within each division that are similar, but we name them differently. So in the central nervous system, a bundle of neuron fibers, which is really the axon of a neuron that gets wrapped in a connective tissue, we call this a tract when it is within the brain or the spine. That same type of structure, axons wrapped in connective tissue, when in the peripheral nervous system, we call them nerves. A concentration of neuron cell bodies within the central nervous system we call basal nuclei. When it is a knot-like swelling in a nerve, which means in the peripheral nervous system, that also contains neuron cell bodies, we will call these ganglion. So in this picture, hopefully you can see the differences in the color. The pink areas are the central nervous system. Notice the brain, and the extension of the brain is the spine. Also notice the spine actually ends where you wouldn't expect it, I would think. Kind of mid-thorax, almost. Not really, it's a little bit lower than that. But you can tell in this picture that it doesn't go the whole way down like to the end of the buttocks, technically. Now, when we're looking at the peripheral nervous system, that's the nerves in yellow, and we can see the little bulb structures along the spine and below it, along the nerves that continue on below it, that are called the ganglia. When we go into the peripheral nervous system, then we have to start dividing it further. The peripheral nervous system will be divided into two divisions, a sensory or afferent division and a motor or efferent division. Listen to the words. If it's sensory, it's sensing information or picking up stimuli and bringing it into the body or into the central nervous system. It's going to do that so that it can interpret the message and then send it out specifically. The somatic sensory division will pull information from the muscles, the bones, the joints, and the skin, and pull it into the central nervous system, whereas the visceral sensory system will pull information towards the central nervous system from the organs, from your guts, from the heart, the lungs, stomach, bladder, etc. When we get into the motor division of the peripheral nervous system, or the efferent with the letter E, this one is responsible for the action or the response. This division is what makes us move or speak or behave. An effector is a cellular organ that will respond to the command of the central nervous system or to the command of this motor division. So within the motor division of the PNS, we break that down further. We have a somatic motor division, and this is primarily responsible for skeletal muscle movement, but also somatic reflexes, or our involuntary muscle contractions. When we look at the visceral motor division of the peripheral nervous system, we also call this the autonomic nervous system. This is responsible for carrying signals to our glands, our cardiac muscle, and smooth muscles. So this is the involuntary portion of us. This one will gather information and then dictate or direct an action or behavior of your organ systems, your smooth muscle, and your heart. Within this division, then we break it down even further into the sympathetic division and parasympathetic division. The sympathetic division of the autonomic nervous system, this is your fight or flight response. This is, are you going to fight the tiger, run from the tiger, freeze, or fawn? This gets the body ready for the interaction. This part of the nervous system will deliver an organ, such as the heart. We'll talk about the heart, the ability to beat faster. You will breathe faster when this part of the nervous system is hypertoned. But this part of the nervous system will also inhibit digestion and urinary functions. So I want you to think about that. If you had to decide if you were going to run or fight from a tiger, your body's concern after evaluating the environment and the situation is either to run or fight. So it will not care about digesting food as it's getting your body ready. It's not going to care in that minute about purifying your blood. Now, the parasympathetic division is what we would call our wine or dine response. It's when we are truly able to relax. It is when we are out of that stressful situation. We don't have to decide if we're running or fighting the tiger. It is when we're able to allow our body to heal. When this happens, when the parasympathetic division is allowed hypertone, it will calm us, slow our heart rate and breathing, and stimulate the digestion and urinary systems. So if you need a picture, this next little chart is really helpful for that. You can walk through the pieces and the parts. Again, central nervous system broken into brain and spine. Pretty simple. Just a little bit of nomenclature difference when we're talking CNS in comparison to PNS. The PNS then divides itself into sensory or motor divisions. Within the sensory division, again, the only job that division has is to evaluate the environment, pull in the stimulus toward the central nervous system so it can figure out what's happening and where it is. So it has a visceral sensory division and a somatic sensory division. Again, visceral tells us organ system, what they're experiencing, what they're going through. Somatic, that's body wall stuff. When we talk about the motor division, motor, think of it as the motor goes into action or moves us. So within the motor division, we break that into visceral motor, which means organs and our guts, the motor division of that part, what makes them do what they have to do. And then the somatic motor division. Somatic motor division, again, that is going to be what the skeletal muscles are up to and any reflexes that might be activated. When we talk visceral motor system, how our nervous system directs our heart to beat, our bladder to do its thing, our intestines to do their thing, our vessels to do their thing. That is under the direction of this portion of the nervous system. And we call this also the autonomic nervous system. It is involuntary. It's taking stimuli in, evaluating it, and directing it. In this system, we have the parasympathetic division as well as the sympathetic division. Parasympathetic is relaxing, rest and digest, wine and dine. We get to filter blood and urinate properly. We get to eat and digest our food well. Also, there are going to be some reproductive features that work better under parasympathetic tone. In the sympathetic division, this is fight or flight stuff. Are we running or are we fighting? And it is prepping the body for either one of those. So what it is concerned with will be an accelerated heart rate, accelerated breathing rate, but it's not worried about digesting the food at that point or filtering blood at that point. When we talk about neurons, we have to talk about excitability or irritability. A neuron has to be able to respond to environmental changes, that is what we're calling a stimuli. It has to be able to conduct electricity, like we learned about previously about action potentials and thresholds and all of the things that happen across the plasma membrane to instate that polarity difference from one side of the cell membrane to the other. Neurons will be able to secrete things. When each signal reaches the end of a nerve fiber, this is when they secrete neurotransmitters. Remember, these are chemical messenger packets of information. These are the codes. They get secreted across the gap, which then will be received by the next cell in line or the tissue or some other type of effector or receiver. When we talk about the functional types of neurons, what that means is this. We learned about the divisions in the nervous system, so really they relate. We will have neurons that are considered sensory afferent neurons. That is what they do. Their job is to pick up information, detect the stimuli, and transmit it to the central nervous system. These are called afferent neurons with an A. Then we can have what's called an interneuron or an association neuron. These are like the ones in between. That's why it's called an interneuron. An interneuron we will find only in the CNS. And these are this is the bulk of the neurons in the CNS will be an interneuron. Because the CNS, remember, is the point where we take the information and we figure out what it means and then what we're going to do about it. These are the ones responsible for processing information and making decisions. A motor neuron or an efferent neuron with an E This will send out a signal to a muscle or a gland or an effector. It's called a motor neuron most of the time because it will have an impact on the muscle. Now, this could be skeletal, this could be cardiac, this could be smooth. But it also can go to glandular tissue. An efferent neuron will conduct the signal away from the central nervous system. So the motor neuron does the job after it's been commanded by the central nervous system, after the central nervous system has done its decision making. So if this picture might help you a little bit, you can see that, okay, so we'll pretend you have an afferent nerve in your finger and you touch a hot stove. That information or that stimulus will be transmitted to your central nervous system, where we will find the interneuron or the association neuron. Again, we only find them in the CNS. Then that will connect with a motor or efferent neuron that will conduct a signal from the central nervous system to an effector such as a muscle or a gland. When we look at the structure of a neuron, we have to talk about some common parts that we will see. And even after we find the common parts, there are going to be some different shapes of neurons that we'll explore. So the soma, this is the cell body, also called the neurosoma or the perikaryon. It has a centrally located nucleus with a large nucleolus. Within the cytoplasm, we will find mitochondria and lysosomes and a Golgi complex, numerous inclusions, and an extensive, rough endoplasmic reticulum and cytoskeleton. Within the cell body of a neuron, we're not going to find centrioles because they don't reproduce. As far as regenerating a neuron, that has been shown to be able to happen. It's not easy and it may take time, but there is potential for that to happen. Now on this picture, we can see a couple other things here. Off of the cell body, we see this area. It's kind of like a triangle. It's called the axon hillock. And then you have this tail that comes off of the neuron. This is its axon. You only have one axon and it might have some collateral fibers coming off of it, as in this case, we have an axon collateral that comes off of the main axon. Now off of the cell body, we notice these little branches. These are the dendrites. The dendrites are responsible for the reception of information or stimuli or whatever, whereas the axon is responsible for the sending out or perpetuation or continuation of maybe an electrical impulse. At the very end of the axon, we notice these little branches. This is the terminal arborization. The word arbor should tell you it's going to look like a tree. At the end of these little branches, this is where we have these synaptic knobs. Remember, we learned about the synaptic knob already. One thing that is interesting to note, the more dendrites that a neuron will have, well, it kind of makes sense too, the more information it can receive and process. Again, we'll have one axon per neuron. They can be cylindrical and unbranched for most of the length, but sometimes, again, they might have a collateral coming off of it. The axoplasm is the name we give for cytoplasm that is contained within the axon. The axolemma, this is the plasma membrane of the axon itself. A Schwann cell is what is responsible for the production of the myelin sheath that will enclose the axon. And this helps with conductivity of electrical potentials. When we look at different structures of neurons. We can have a multipolar neuron. We may have one axon with multiple dendrites. This is the most common. Most neurons in the brain and spinal cord will be multipolar neurons. And if you think about the point of the neurons within the central nervous system, again, that is major integration center, then that makes sense. You'll get a lot of information pulled into that portion of things. A bipolar neuron. This is one axon and one dendrite. We will find bipolar neurons in olfactory cells, in the retina, and in the inner ear. So you're noticing bipolar neuron will be found in areas where we have special senses, where it's needs more specificity and fine-tunement. A unipolar neuron, this has a single process leading away from the soma. And then we can find a dendrite on one end and an axon at the other end. We'll find this in skin and organs, maybe leading up to the spinal cord. And an axonic neuron, it has many dendrites, but in this case, this has no axon. And this is interesting because it helps in visual processing. If you think about vision, really what it does is just sense information. It is a special sense. So it pulls information in. When we talk neuroglial cells, and we mentioned this very briefly, or I did when we went through tissue identification, there are about 1 trillion neurons in the nervous system. Neuroglia or glial cells will outnumber neurons by about 50 to 1. That is because their role is to support and protect the neuron. They can bind neurons together, form a framework for nervous tissue. In the fetus, neuroglial cells are known to guide migrating neurons to their destination. So they're like little escorts and shuttles. And they can give precision to a conduction pathway. Now, we recognize 6 different types of neuroglial cells. 4 of them occur only in the brain and spine, in the central nervous system. Those 4 types are oligodendrocytes, ependymal cells, microglia, and astrocytes. So an oligodendrocyte in the central nervous system is responsible for the formation of that myelin sheath. They each will have an arm-like process that wraps around a nerve fiber, forming an insulating layer that will speed up the signal conduction. An ependymal cell will line the internal brain cavities. This is primarily cuboidal epithelium with cilia on the apical surface. The purpose of these ependymal cells is to secrete and circulate CSF. We will find microglia. These are macrophages formed from white blood cells that look for cellular debris. The astrocyte is an interesting neuroglial cell within the CNS that helps form a framework for the tissue. This is also going to be one of the primary formers of the blood-brain barrier. Nerve growth factors may be secreted by astrocytes to promote neuron growth, as well as synapse formation. When a neuron becomes damaged, the astrocytes will form hardened scar tissue around it and fill the space formerly occupied by the neuron. This is what the astrocytes are doing. The two types of neuroglial cells that we're going to find in the PNS then are Schwann cells and satellite cells. So Schwann cells will envelop the nerve fiber and wind around it repeatedly. These Schwann cells are responsible for the production of the myelin sheath, similar to the oligodendrocyte in the CNS. So then the satellite cells that we find in the PNS, these will surround the neurosomas in the ganglia. They provide insulation around the soma and help regulate the chemical environment of the neurons. So this picture is really great. So here are neuroglial cells of the central nervous system. Here we get a great picture of that astrocyte, how it looks like. Again, it's one of those sticky things that you throw out the window. But this will help form the framework. And again, if there are damaged cells, it will sclerose and form scar tissue. Here is a picture of those ependymal cells, again, responsible for the secretion of CSF, so super functional and important. They're all super functional and important. Here's an example of the oligodendrocyte. Here's the cell itself. It will have an extension that will wrap around differing axons and creating the myelin sheath within it. And then we have the macrophages, which are wandering around taking care of debris. So we've mentioned myelin multiple times. Let's talk about what it is. So myelin is the insulating layer around the nerve fiber. It is produced by the oligodendrocyte within the CNS and then the Schwann cell in the PNS. It consists of the plasma membrane of glial cells. When we talk about myelination, then this is the production of the sheath. It will begin in the 14th week of fetal development and proceed through infancy. It is completed in late adolescence. In order to have the body produce myelin appropriately, dietary fat is hugely important to do the development of the nervous system and to maintain it. In the PNS, the Schwann cell spirals repeatedly around a single nerve fiber. It will lay down as many as 100 layers of its own membrane. No cytoplasm between the membranes. So again, it's just cell membrane layers and layers of cell membrane. It produces what's called the neurolemma then, which is the outermost coil of the sheath that will contain a nucleus and cytoplasm. The neurolemma is external to the neurolemma is a thin layer of fibrous connective tissue that we call the endoneurium. In the CNS, we noticed on that picture that we just looked at a couple slides ago that the oligodendrocytes will reach out to myelinate many fibers, whereas in the PNS, the Schwann cells, it's specific to one. The nerve fibers in the CNS have no neurolemma or endoneurium. So that's another contrasting difference. So here is how myelination then will happen within the peripheral nervous system. So there's our axon, there's our Schwann cell. Remember, it is just multi-layers of plasma membrane until eventually it isn't. So the endoneurium we get to see on the very last part of the picture here is that outer wrapping paper around the sheath. we get to see the nucleus and the cytoplasm that was responsible for creating the role of, really, it's a role of phospholipids. Here in this cross section, we get to see a better representation of how that all lines up. And then contrasting the myelination in the CNS. Again, Schwann cells, peripheral nervous system, they're specific to an axon, whereas the oligodendrocyte in the CNS will attach to many different axons, producing the myelin, but it won't have the endoneurium on it. So this will lead us into then the conversation of multiple sclerosis. You've heard of MS. It's a degenerative disorder of the myelin sheath. The oligodendrocytes and myelin sheaths in the CNS begin to deteriorate. What happens is it's sclerosis. It gets replaced with that hardened scar tissue. So something attacks, and it is classified as autoimmune with a genetic predisposition. But remember, epigenetically, okay, there's a lot to that. You can have a stressful triggering event that can send someone with a predisposition into a spin for sure. But remember, hard genetics, 20% of what we manifest, 80% of what we're doing. So what happens when the scar tissue builds? The nerve conduction doesn't happen correctly. So this is when a person may get double vision and tremors and numbness and speech deficits, but also trouble walking. And humidity tends to be really hard for a person with MS. Temperature stresses on the body. And clearly humidity is not only temperature, it's moisture, right? So the typical onset is between the ages of 20 and 40. It can be fatal from 25 to 30 years after diagnosis. Again, classified as autoimmune, which tells us hypersensitivity response of the immune system, a self-attack, triggered by a viral infection, which there is a strong correlation to viral triggers to autoimmunity. And I mentioned before, correlations to viruses and cancer as well. There is a woman, and I want to say her name is Dr. Terry Wall, and she does TED Talks, and she is a medical practitioner who had MS be displayed and show up for her. What she did was she went into she was non-ambulatory from what I recall from her story. And she said she had enough of it and she went into her medical stuff and she learned what she needed to do to make things different. And in a relatively short period of time, she was ambulatory again. So it's a great story. And I think that if you know anybody with MS, I love stories of hope that makes us keep trying and keep doing things. Okay, so let's pop into the next section. We're going to look at nerve conduction velocity. This is where we're going to get a little bit thick into the information. We're going to look at the gated ion channels in regards to membrane potentials. We're going to look at voltage gated ions and ligand gated ions. We're going to review resting membrane potential. We're going to review depolarization, repolarization, and even hyperpolarization. We're going to look at gated ion channels that are essential for the development of an action potential. We're going to look at the sequence of events that lead to the generation of an action potential. And we're going to look at ionic movements that result in changes to the membrane potential and sodium-potassium exchange in maintaining the potential. So when we talk about the conduction speed of the nerve fiber, It will be dependent on two factors: the diameter of the fiber and the presence or absence of the myelin. The signal conduction occurs along the surface of a fiber. Large fibers will conduct signals more rapidly because they have a greater surface area to do so. The purpose of myelin is that it will further the speed of signal conduction because it insulates the axon. So if anyone knows anything about electricity, if you have an uninsulated wire versus an insulated wire, it's like a containment principle almost. You keep the things moving down the axon, okay, kind of contained in the tube, but where they have little spaces and separations, it's almost like the action potential can hop or jump. So conduction speed, if we have a small unmyelinated fiber, 0.5 to 2 meters per second, the conduction can travel. If we have small myelinated fibers, it can go from 3 to 15 meters per second. If it's a large myelinated fiber, it can conduct the potential up to 120 meters per second. So we have then, if you understand how this stuff may work, slower signals will supply the stomach and dilate the pupil where speed is less of an issue. We will have it set up for faster signals to supply skeletal muscles and transport sensory signals for vision and balance. Now, resting membrane potential. Remember, it's the charge difference across the plasma membrane. -70 millivolts is what we like to have in a resting unstimulated neuron. Remember that the negative value means there are more negative particles on the inside of the membrane for various reasons. The resting membrane potential will exist because of an unequal electrolyte distribution between the ECF and the ICF. The resting membrane potential will result from the combined effect of 3 factors: ions that diffuse down their gradient, the plasma membrane being selectively permeable, and then the electrical attraction of cations and anions to each other. So when one thing moves, something else wants to move right along with it. Cytoplasmic anions cannot escape, maybe due to the size or charge. So that might be, RNA or ATP or proteins. The membrane is much less permeable to high concentrations of sodium found outside the cell when it's at rest. The sodium potassium pump pumps out 3 sodiums for every 2 potassiums that it brings in. It will work continuously and require a great deal of ATP. This will necessitate glucose and oxygen being supplied to nerve tissue. So if you need a picture to help understand that, here it is. When we talk about sodium potassium pump, we can reference the ECF and the ICF. If we have a potassium channel and a sodium channel, the sodium concentrated outside of the cell in the ECF sets up the action potential. We might have, again, here we have evidence of larger ions that can't escape, and we have more potassium on the inside of the cell. Now, we mentioned this, I believe, in the last chapter, that we might have some local potentials, disturbances in the membrane when a neuron gets stimulated. It can happen, locally, but it has to reach a threshold to continue it down the length of the membrane. Neuron response begins at the dendrite, which then spreads through the soma and traveling down the axon. It will end at the synaptic knob. When a neuron is stimulated, and this could be by a chemical, by light, by heat, or some type of mechanical disturbance, this is how things will go. It will open those sodium gates that allow sodium into the cell. The inflow will neutralize some of that negative charge. Remember, starting out at negative 70 millivolts. Then the voltage measured across that membrane will start neutralizing itself and coming to neutrality. This is what we call depolarization. It's coming into a neutral state. The sodium diffuses for short distance on the inside of the plasma membrane, which will produce a current that will travel towards the cell's trigger zone. If you remember on the picture of the neuron, right above or right near that axon hillock where the axon comes out of the neurosoma, that's the trigger zone. This short range change in voltage is called a local potential. So here's an axon, and here's a picture of it. Here are dendrites. They get stimulated by something. The change in potential will travel into the soma. If it's enough to make it to the trigger zone, it will be carried down the length of the axon. The action potential, again, this is a more dramatic change produced by voltage regulated ion gates in the plasma membrane. This will occur if local potential from the dendrites and the soma reach a threshold voltage in the trigger zone. The trigger zone then is that area of the axon where the action potential is generated. If excitatory local potential spreads all the way to the trigger zone and is still strong enough when it arrives, it can open these gates and generate an action potential through the rest of the neuron. When we talk about action potentials, we have to talk about that threshold. This is the critical voltage to which local potentials must rise to open the voltage regulated gates. And this will happen around negative 55 millivolts. So the action potential is a rapid up and down shift in the membrane voltage. Sodium ions will arrive at the hillock and depolarize the membrane at that point. When we have the threshold reached, that negative 55 millivolts, this is when we're going to see the neuron fire and the action potential will be carried. So more and more sodium channels then will respond by opening in the trigger zone in a positive feedback cycle, which will create a rapid rise in the membrane voltage, or what we would call a spike. So when the rising membrane potential passes zero, the sodium gates are inactivated and they'll start to close. When they're all closed, the voltage will peak at about positive 35 millivolts. The membrane is now positive on the inside and negative on the outside. Polarity reversed from the RMP is depolarization. By the time the voltage peaks, there are slow potassium gates that will be fully opened. The potassium will be repelled by positive intracellular fluid that now exits the cell. And this outflow of potassium will repolarize that membrane. Remember, depolarization is when we're coming to neutral. When we repolarize, we're reinstating that difference that we want, that we started with. So the potassium gates will stay open longer than the sodium gates did. So slightly more potassium will leave the cell than sodium entering. This will drop the membrane voltage 1 or 2 millivolts more negative than the original RMP. We call this overshoot hyperpolarization or an after potential, and it will regulate itself. Some will leak out. This is good news because then action potential can be reset very quickly. So here's an example. If you need a chart to help your eyes, this will help. Here's our negative 70 millivolts. We're resting. Some stimulus starts to fire or change the electrical charge of ICF to ECF. We hit that negative 55 millivolts, which is threshold. The depolarization begins to happen. Once it reaches a positive 35, the sodium channels will shut down. Repolarization happens. So we can reinstate that negative charge potential on the inside of the cell. Again, that hyperpolarization is kind of an overshoot, but that's okay. The gates will allow some sodium kind of to move in and out, and then we will resume our regular action potential. If threshold is not reached, it will not fire. That's really important to understand. So you can have a lot of little local potentials, but if the negativity on the inside of that cell does not get reached for whatever reason, it's not going to do its thing. So when we're talking about this, we have to understand there's something called a refractory period. During an action potential and a few milliseconds after, it's difficult or impossible to stimulate that region of a neuron to fire again, and that should make sense. It will take linear time to reinstate the polarity principles. If the polarity is not what it needs to be to fire, there's nothing there to do the work. So 2 phases of the refractory period is the absolute refractory period, when no stimulus of any strength will trigger that action potential. If we have a relative refractory period, only especially strong stimulus will be able to trigger a new action potential. And that might be because it has further to go. Okay, so when we talk about a synapse, let me see where we are here. Okay, we talked about these before. They're a neat setup. A nerve signal can go no further when it reaches the end of the axon. It's done. It's made its way through. It triggers the release of that neurotransmitter. This will stimulate a new wave of activity in the next cell, because the next cell will be able to receive that neurotransmitter across the synaptic cleft and be inspired to do things within its own cell. So the synapse between 2 neurons. The first neuron in this signal path is called the presynaptic neuron. That should make sense. This is the one that will release the neurotransmitter. The second neuron in the lineup is the postsynaptic neuron. This is the one that receives and responds to the neurotransmitter. The presynaptic neuron may synapse with the dendrite, soma, or axon of the postsynaptic neuron to form different types of synapses. There's an axodendritic, axosomatic, or axoaxonic synapse. The neuron can have an enormous number of synapses. Like spinal motor neurons will cover or be covered by about 10,000 synaptic knobs from other neurons. In the cerebellum of the brain, one neuron can have as many as 100,000 synapses. So let's just put those words into pictures. In the first picture, we have picture A is showing us the synapse between a presynaptic neuron and a postsynaptic neuron. Again, just look at the words. Presynapse is the neuron before they join. Postsynaptic is the neuron after they join. The synapse, again, is their joining point. So when we have then, looking at picture B, an axodendritic type of synapse, we have the axon of the presynaptic neuron synapsing with the dendrite of the postsynaptic neuron. An axosomatic synapse then would follow suit in the nomenclature. The axon of the presynaptic neuron will synapse with the neurosoma or the neurocell body. Then an axoaxonic synapse will have the presynaptic neuron synapse with the axon of the postsynaptic neuron. So in this picture, I love pictures like this, by the way. This is courtesy of McGraw Hill, these pictures, so thank you. Also, Omicron Science clearly gave us this picture. So super cool. We get to see synaptic knobs and how they will synapse with the cell body, in this case, the soma of the postsynaptic neuron. So all the little knobs you see would be from those terminal branches of the presynaptic neuron anchoring into the neuron or the neurosoma of the postsynaptic neuron. Super cool. This picture should look familiar to you because we covered this in our previous lecture when we were talking about the neuromuscular junction and their similarity in the concept. So the structure of a chemical synapse. Here we have our axon. Within the axon, again, we will have the axoplasm. It will have microtubules of the cytoskeleton, mitochondria all through it. When we get to the axon terminal, or the knob, the synaptic knob of the terminal, it will at that point have inspired a production of neurotransmitters and a correspondent release inspired by that action potential. The neurotransmitters will be released into the synaptic cleft and received then by the postsynaptic neuron. Now think through this for a second here. All of this has to work in order for everything to fire appropriately. So from electrical conductivity principle of the presynaptic neuron to the production of the appropriate neurotransmitter in an appropriate quantity, the release of the neurotransmitter into the cleft, and then on the postsynaptic neuron, having the appropriate receptor sites for that specific receptor or neurotransmitter to be received. So in this picture also, we see on the postsynaptic neuron, the receptors are indicated in red. And also we're going to have what's called a ligand regulated ion channel, so specific to a specific chemical. So let's talk about neurotransmitters. They're super cool. There are tons that could be present in our bodies. They have different classifications according to their structure and according to their function. If we don't have the resources to build them, we won't. So I think this is a really relevant topic as we're talking about the impact of this kind of stuff on overall physiology, action, things that the body is able and capable of doing. The neurotransmitter will be synthesized by that presynaptic neuron. So again, everything in that cell has to be working in order for the neurotransmitter to even be produced. They are released in response to some type of stimulation. In this case, we are talking about the action potential or an electrical gradient or pulse. Then they have to bind to specific receptors on the postsynaptic cell. So even if a particular neurotransmitter is produced, if there are no receptors for it, it won't be received. It will be broken down, degraded, and reabsorbed, hopefully. A given neurotransmitter does not have the same effect everywhere in the body. This is a good concept to grasp. We'll see this again when we get into endocrine system. Just because it's produced and released, if we do not have the capability of hearing the message, we can't hear the message. So what this is saying is only certain cells will have certain receptors for either hormones or neurotransmitters or whatever it is. And in this case, we're talking through neurotransmitters. Multiple receptor types can exist for a particular neurotransmitter. For example, there are 14 receptor types for serotonin, meaning that serotonin can be released, and you have to have a particular type of receptor, again, in order to be able to receive the message. And then depending on what kind of receptor it is, will have a downstream impact on the effectiveness of the neurotransmitter or what it's able to do. The receptor governs the effect that the neurotransmitter has on a target cell. This is according to which type of ion channel opens. So the production is one thing, the reception is a bigger deal. Neurotransmitters are diverse in what they are capable of performing. Some have an excitatory mechanism, and some will have an inhibitory style of effect. The effect depends on the kind of receptor the postsynaptic cell has. So same neurotransmitter can be released by two different cells. Presynaptically, the postsynaptic cell will be the dictator of what that neurotransmitter will do, dependent upon the specific receptor in the postsynaptic cell. So if the receptor opens up ion channels that let in positive ions, threshold can likely be reached. However, if the receptor opens up channels that let negative ions into the cell, threshold therefore will be harder to reach. So when we're talking neurotransmitters and the kind of synapses and how that contributes to how they are capable of acting, we will recognize excitatory cholinergic synapses, excuse me, inhibitory GABAergic synapses, and excitatory adrenergic synapses. A cholinergic synapse. Acetylcholine will be the neurotransmitter involved in this type of synaptic connection. The acetylcholine will excite the postsynaptic cell, but inhibit others. It will excite skeletal muscle, but inhibit cardiac muscle. If it has an excitatory action, what happens in that case, in the case of skeletal muscle, the acetylcholine will diffuse across the cleft. It will open ligand-regulated sodium gates on the postsynaptic neuron, whereby sodium will enter the cell and depolarize it if enough enters. If the threshold is reached, then voltage-regulated ion gates open in the trigger zone, causing that postsynaptic neuron the capability of firing. So here's an example in pictures of what we just walked through. Here's our presynaptic neuron. And we're talking about an excitatory cholinergic synapse, delivering the availability of an action potential to be transferred into that postsynaptic neuron. If it's an inhibitory synapse, it's GABAergic, inhibitory GABAergic synapse. First of all, GABA, gamma aminobutyric acid. This type of synapse will employ gamma aminobutyric acid as the neurotransmitter. The nerve signal will trigger the release of GABA into the cleft. And the GABA receptors are chloride ion channels. So the chloride will enter the cell, making the inside more negative. Okay, remember, and we're starting at around a negative 70 millivolt charge difference or resting membrane potential. So if we're bringing chloride into the cell, we're making that inner world already more negative by the presence of the chloride. If we do that, it's going to be harder to depolarize the cell. So this is why it's part of the reason it's inhibitory. It will, that postsynaptic cell then will be less likely to fire. Let me pause for a second here and talk about gamma amino butyric acid in relation to what that equates to in our body, just for a little connection point here. When we are deficient in GABA, there will be a tendency toward anxiety. So a lot of these neurotransmitters, yes, they just make physiology happen, but there are several that we have linked to psychology as well, which I think is fascinating, right? So we have this biochemical foundation and understanding too of either overload or deficit. And then we can even beyond that connect the neurotransmitters deficit or overabundance to brainwave activity. So it's a really cool interaction between all of the things. So the cessation of a signal, this is when we stop adding neurotransmitter and getting rid of what's there. When this happens, this will stop the signals in the presynaptic nerve fiber. And what happens, we had mentioned this back in the other chapter when we were talking about the NMJ, about how the neurotransmitter can be broken down and reabsorbed. Okay, these are ways that this can happen. So diffusion can take care of the signal stopping. The neurotransmitter will escape the synapse, maybe into the nearby ECF, where astrocytes in the central nervous system can reabsorb it and return it to neurons. Reuptake, where the synaptic knob reabsorbs some of the neurotransmitters, and that is what we touched on previously. Also, a little bit of degradation in that cleft. We touched on that as well. Enzymes that may be present in the cleft can help degrade the neurotransmitter and then again reabsorbed back into the synaptic cleft. This is relevant when we talk about some medications that we take for mood disorders. So, an SSRI is a selective serotonin reuptake inhibitor because we don't want the reuptake or the degradation at that time. So what's the purpose of a synapse, could we say? Synaptic delay will slow the transmission of nerve signals. More synapses in the neural pathway, the longer it takes for information to get from its origin to destination. I don't think that is difficult to understand. But we feel at this point, the theory, the hypothesis and the relevant evidence, I would say, is that it helps us to process information, to store information, and actually in decision making. So the synapse allows for information integration rather than just reaction principles. Synapses are the decision-making devices of our nervous system. And I would say along with that interneuron exchange that we learned about, the interneurons being part of the central nervous system for interpretation, integration, and then, choice of action and or behavior. The more synapses that a neuron has, the greater its information processing capabilities. We have something called a pyramidal cell in the cerebral cortex, and they have about 40,000 synaptic contacts with other neurons. The cerebral cortex in our brain, this is our main information processing tissue. It has an estimated 100 trillion synapses. Neural integration, that's the ability of our neurons to process, store, and recall information all so that we can make a decision. And that's kind of what makes us very cognitive beings. Summation. We mentioned summation when we were going through the last chapter, and we were talking through, stimulus and threshold, and then how it sums up, and the stimulus must reach threshold. So one neuron can receive input from thousands of other neurons. Summation, when we say that in regards to this topic, this is the process of adding up stimuli and responding to the net effect. This enables the nervous system then to make decisions. So we have a couple types of summation. We have temporal summation. This occurs when a single synapse receives stimuli so quickly that an effect accumulates. Ions will add up over time to a threshold voltage that triggers an action potential. Spatial summation, this occurs when stimuli from several different synapses will add on top of each other. So several synapses can emit enough sodium to reach threshold. A presynaptic neuron will cooperate to induce the postsynaptic neuron to fire. So this picture shows us that very well, I think. So the first picture, picture A, is temporal summation, where we have synapses joining right at the neurosoma of a postsynaptic neuron. So we have an intensity variation through one synapse. If we have all three of the presynaptic neurons delivering some type of stimulus to the neurosoma, it can oftentimes be enough to reach that threshold, take it to the trigger zone, and fire the postsynaptic neuron as well. So either the intensity has to be really high of stimulus, or it can be 3 weaker stimuli or multiple weaker stimuli that add up to equal enough change in the resting membrane potential to fire that postsynaptic neuron. When we talk about inhibition, the word tells us something will be stopped. So presynaptic inhibition, this is the process in which one presynaptic neuron will suppress the other one. So it will reduce or halt unwanted synaptic transmissions. An inhibitory neuron will release inhibitory GABA. This will prevent channels from opening in the synaptic knob, and the presynaptic neuron release less or no neurotransmitter. Therefore, the signal is inhibited. And that's how this type of synapse really works. So let's touch on some of the pathologies. Okay, Alzheimer's disease. Alzheimer's disease is a very interesting diagnosis of neurodegenerative diseases. What I've noticed is that in Alzheimer's disease, the physical body is maintained, like there are no health problems, but there is no cognitive ability any longer, where functionality just starts to be lost. So with Alzheimer's disease, and there are a ton of theories on how this gets created, it has to do with a certain type of plaquing. It's called a beta amyloid protein that plaques the brain, and it almost does create, from what I understand, a scar tissue like representation within the brain. Now, Alzheimer's is actually under the umbrella of dementia. There are other kinds, and the true diagnosis, also from my understanding of Alzheimer's, cannot be delivered until autopsy. It is responsible for about 100,000 deaths per year. 11% of the population over 65 will manifest this. 47% of people by the age of 85 are being known to have this now. There's a lot of speculation why this is such a big thing. Memory loss. So short-term memory is gone. Okay, moody, sometimes combative. They lose their ability to talk, walk, and eat. When they lose their ability to eat, there's not much left to do or that can be done. Now, they have medications that may be able to diminish symptoms a little bit, and but the prolonging of the symptoms and the vast deficit that comes, the medications that are available right now, they don't hold it off very long and all that well. So this is a tough road, and care for an Alzheimer's patient is very difficult. They have shown to show deficiencies of acetylcholine and nerve growth factor. I would go here and also say there is a potential dopamine deficiency, maybe, or something is not working with dopamine. What happens is there is an atrophy of the gyri, or the folds in the cerebral cortex. There is a tendency of a genetic predisposition, as with many things. And the treatments aren't great. You can give nerve growth factor or cholinesterase inhibitors. Now, recently I just watched a TED Talk about medium chain triglycerides, fatty acids in the diet, that have shown to be beneficial for cognitive decline and neurodegenerative diseases. And there are products available to help with this. Excuse me. But coconut oil, from my understanding, is one of the foods on the top of the list that can help with this. Okay, so here is a picture of a brain. So the gyri, again, those are the folds, the kind of the noodles. The grooves, those are sulci. And you can see on the microscopy picture where there's evidence of plaquing. Another neurodegenerative disease that we have to touch on is Parkinson's disease. There's a progressive loss of motor function, generally beginning in the 50s or 60s, and no recovery that we're aware of. They're doing some implants, I think, implant devices like electrical implant devices that I want to say I have read are helpful in some cases. There is a degeneration of dopamine releasing neurons in this case, and there's a particular center in the brain that would be responsible, or the primary area of deficit. Dopamine normally prevents excessive activity in motor centers or the basal nuclei in the brain. So this disease will cause involuntary muscle contractions, something that we call a pill rolling motion, facial rigidity, and slurred speech. Handwriting becomes ineligible, and the gait slows down, and they might shuffle. I want to say that this isn't on this slide, but I really do think there's a higher propensity for this to happen in males more than females. The treatment thus far, like I said, I want to say they've come up with a device that gets implanted in the basal nuclei that may be able to help with this. Generally, drugs and physical therapy. A dopamine precursor called L-DOPA has the ability to cross the blood-brain barrier, but it does have bad side effects on the heart and the liver. Also, MAO inhibitor will slow neurodegeneration. And if there's a tumor, there may be some surgical procedures that can be done as well. Okay, so that's it for this lecture, and we'll see you in the next one.